we're looking at nucleotides.
Right.
Specifically, the metabolism of purines and pyrimidine.
And for everyone listening who just heard purines and pyrimidines and immediately thought about skipping ahead,
Don't do it.
don't do it. Stick with us. Because this is not just some dry chemistry lecture. This is the story of how your body builds your genetic code, your DNA, your RNA, from absolute scratch.
And not just that, it's how it mints the energy currency of the cell.
ATP, the stuff that's keeping your heart beating right this second.
It's the cell's operating system, really.
So our mission today is to decode that chapter, but not just read it. We want to understand the why and the how. How does the body actually manufacture these critical building blocks?
And how does it regulate that assembly line? So it doesn't just spin out of control.
And this is the part that, frankly, terrifies me. What happens when the machinery breaks? Because when this machinery breaks, it's not a minor glitch. We're talking about crystals forming in your joints.
Yeah, that's gout.
Total immune system collapse. And some really, really tragic neurological conditions where the brain basically turns on itself.
It's an incredibly high-stakes game.
Yeah.
And on the other side of that coin, understanding this machinery is precisely how we've developed some of our most powerful weapons against cancer.
Right, because you can target it.
You can target it. So, yeah, the stakes could not be higher.
Let's jump right into the big picture then. The first thing that really stopped me in my tracks when I was reading the introduction was what I'm calling the paradox of diet.
Purines and pyrimidines from your diet are generally not incorporated directly into your tissue nucleic acids.
That seems, insanely wasteful to me. Evolution usually pinches every single penny. If there's a calorie to be saved, the body saves it. So why throw away perfectly good Lego bricks?
Well, you have to think about what DNA is. It isn't just a brick. It's information. It's code. If you started integrating the DNA from the cow you ate, or from bacteria in that salad, or from a virus, you are risking genetic corruption.
So you're risking putting foreign instructions into your operating system.
Exactly. It's a security protocol. Yeah. The body has this scorched earth policy. When you eat nucleoproteins, your digestive tract just goes to town.
It just destroys them.
It degrades them into mononucleotides, then into free bases, and for the most part, those purines get oxidized into uric acid, and you excrete them.
They're gone.
Gone. The pyrimidines are broken down into other stuff. The point is, we destroy the foreign code completely. The body prioritizes genetic integrity over energy efficiency.
So if I can't use the letters from the food I eat to write my own genetic code, then I have to write them from scratch, every single one.
Precisely. This is what the text calls the de novo reality. De novo just means from the new.
From scratch.
We have to synthesize our nucleic acids from what are called amphibolic intermediates, which is just a fancy way of saying basic metabolic building blocks.
Like what? What are we talking about?
Simple things. Amino acids, sugar, and even carbon dioxide.
Wait, the stuff I breathe out?
The very same.
So going back to my Lego analogy, it's not just that I'm taking a Lego castle I bought, smashing it into dust, and throwing the gust away.
No, it's more extreme than that.
It's like I'm burning the Lego castle down to ash, and then I'm going out into the backyard to dig up clay and sand to bake my own bricks from scratch.
That is a much, much better analogy. You're changing the fundamental state of the matter. And that process, de novo synthesis, is incredibly complex. And crucially, it's expensive.
Expensive in what sense?
In ATP, the biological currency. It costs a tremendous amount of energy to build these chemical rings from scratch.
And that brings us to the tension of this whole topic. Because this system is so high energy and so vital if it goes wrong.
The consequences are severe. We mentioned gout. That's a painful accumulation of waste products. But then there's something like Lesch-Nyhan syndrome, which is a truly heartbreaking genetic disorder, or adenosine deaminase deficiency, which basically leaves a child with no immune system.
And on the flip side, because cancer cells need to make DNA faster than any other cell.
Exactly. If we understand how the factory works, we can throw a wrench in the gears of the cancer cell. We can block these pathways.
Okay, so let's unpack that factory. Section 1, purine biosynthesis. We're building the double ring. First question. Where is this factory located? Is every cell in my body, sweating over a hot stove making purines?
Not quite. The primary factory, the heavy industry hub of the body, is the liver.
The liver? Okay, wait, hang on. If the liver is the only one making these things, how do they get to my brain? Or my muscles? Or my white blood cells? A purine isn't a tiny molecule.
No, it's not.
Does the liver just ship them out via FedEx?
That's a great question, and it's actually a point of vulnerability. Tissues like the brain, or leukocytes, your white blood cells, They often can't make enough on their own. They don't have the full de novo pathway, or it's not very active.
So they're dependent.
They're dependent. They rely on preformed purines that are transported from the liver, often hitching a ride inside red blood cells. Or, and this is key, they rely very heavily on salvage pathways.
Salvage. Recycling. We'll get to that. So if the liver goes down, the supply chain breaks.
In many ways, yes. Now let's look at the ingredients list for this recipe. Figure 33:1 in our source material gives this beautiful diagram of a purine ring.
It looks like a hexagon fused to a pentagon, a double ring structure.
It is. And it's labeled with where every single atom comes from. It's like a sourced from label on a fancy coffee bag.
It really is a chemical mosaic. Let's walk through it. You've got nitrogen atoms and carbon atoms. Where did the nitrogens come from?
The nitrogens are donated by 3 specific sources. The amino acids, glycine, aspartate, and the amide nitrogen from glutamine.
Okay, let's pause right there on that shopping list. Glycine, aspartate, glutamine, those are all amino acids, the stuff proteins are made of.
Exactly. So we are, in effect, burning protein to make DNA. And look at the cost. Glycine isn't just lending a hand. The entire glycine molecule gets plastered right into the ring.
It's a huge piece of the structure.
A huge piece. It provides both carbons and a nitrogen. This is why protein malnutrition can hit rapidly dividing tissues so hard. If you don't have the amino acids, you can't build the nucleotides, and you can't make new cells.
Okay, so protein provides the nitrogen and some of the carbon. Where does the rest of the carbon come from?
Well, one source is respiratory CO2, just plain old carbon dioxide that you would normally breathe out. The body grabs it and uses it as a building block.
That's amazing.
And finally, you have derivatives of tetrahydrofolate.
Tetrahydrofolate, that's related to folic acid, right? Like what you're told to take during pregnancy.
Yes, that is the active form of folate, and this is exactly why it's so critical for pregnancy. Folic acid is a one-carbon donor.
Meaning?
It's like a tiny delivery truck that carries a single carbon atom to the construction site to complete the purine ring. If you don't have enough folate, you can't close the ring. You can't make DNA.
And if a fetus can't make DNA...
It can't grow properly. That's where you see neural tube defects and other serious issues.
It really connects the dots on why nutrition matters at such a fundamental molecular level. It's a recipe. You need a cup of glutamine, a dash of CO2, a pinch of folate. If you're missing any one of these, the factory just shuts down.
Exactly. And the process itself is a masterclass in biochemistry. It involves 10 distinct steps, but it all starts with the sugar.
Ribose 5-phosphate.
Correct.
Right.
But the sugar needs to be supercharged first. The enzyme PRPP synthetase takes ATP and adds a pyrophosphate group to the ribose.
And that creates the acronym that just kept popping up in the text. PRPP.
PRPP. Phosphorobosyl pyrophosphate. Let's break that down because the jargon sounds scary, but it's just descriptive.
Please do.
Phosphorobosyl just means a ribose sugar with a phosphate on it. Pyrophosphate means 2 phosphates linked together. So it's a sugar that's been loaded up with high-energy phosphate bonds. It's priming the pump.
So PRPP is the foundation. It's the activated scaffold that we're going to build the purine ring on top of.
Precisely.
But the text says making PRPP isn't the committed step yet. Why not?
Because PRPP is used in other pathways, too. It's a general-purpose building supply. You can use it to make pyrimidines, for example. So you haven't decided to build a purine yet. You've just, bought the lumber.
So when do we commit? When do we lay the first stone?
That happens in reaction 2. The enzyme is PRPP glutamyl aminotransferase.
That is a mouthful.
It is.
I practiced it. PRPP glutamyl aminotransferase.
Very good. And the name tells you exactly what it does. It takes an amine nitrogen from glutamine and transfers it onto that PRPP scaffold, kicking off the pyrophosphate group.
And this is it. This is the point of no return.
This is the committed step of purine biosynthesis. Once this reaction happens, the cell is buying a one-way ticket to Purine Town.
And this must be incredibly highly regulated then, right? You don't want to buy that ticket if you don't actually need to take the ride.
Extremely regulated. This enzyme is the gatekeeper. We'll get into the specific breaks in a little bit, but just keep in mind, once the cell pulls this trigger, it is committing substantial energy resources to finish the next 9 steps.
So after we commit, we go through this long assembly line. 10 steps in total. That seems excessive.
It's all about control and specificity. Every step is a potential checkpoint. But here's where human evolution shows off a little bit. In bacteria, each of those 10 steps is usually handled by a separate individual enzyme just floating around in the soup of the cell.
Okay.
But in humans, in eukaryotes, we have...
Multifunctional catalysts. I saw that term.
Yes. We've evolved these massive proteins, multifunctional polypeptides, that contain multiple enzyme activities on a single chain. It's like having 3 or 4 machines bolted together.
And why is that better?
It allows for something called channeling.
Channeling.
Imagine if you were building a car on an assembly line. In the bacterial model, you put a door on in building A, then you have to drive the chassis across town to building B to put the wheels on.
And along the way, the chassis could get lost or rained on or something?
Yeah. Or it might just float away into the cytoplasm. Exactly.
Yeah.
In the human model, the machines are all bolted together in one long line. The robot arm that puts on the door passes the car directly to the robot arm that puts on the wheel.
So the intermediate product never floats away.
It never leaves the enzyme complex. It's protected, it's faster, it prevents these unstable, half-built molecules from getting lost or degrading.
That's incredible efficiency. So nature has actually optimized the factory floor layout over millions of years.
Indeed. So we go through all these steps, we burn a lot of ATP, we add our carbons and nitrogens one by one, and finally we end up with the parent purine nucleotide.
IMP, inosine monophosphate.
That's the one. Now notice, it's not adenine the A or guanine the G yet. It's the prototype.
It's the blank template.
Exactly right. So now we've reached a fork in the road, section 2 in the chapter, from IMP to AMP and GMP.
Okay, this is where the engineering really just blew my mind. The source material highlights a cross-regulation logic here that is just beautiful.
It is an incredibly elegant design. So we have this parent molecule, IMP. We need to turn it into either adenosine monophosphate, AMP, or guanosine monophosphate, GMP.
The A and the G for our DNA and RNA.
Right, but the cell needs roughly equal amounts of both to replicate DNA efficiently. You don't want a million A's and only 10 G's. That would be a complete disaster for genetic coding.
So how does it balance them? Does it have like a little counter on the wall?
It's even smarter than that. It uses energy as the counter. It's a reciprocated engine. To make AMP, the pathway requires GTP as the energy donor.
So to make an A, you need energy from a G.
Correct. And to make GMP, the pathway requires ATP as the energy source.
So to make a G, you need energy from an A.
Yes.
I love that. Let me see if I can put this another way. Imagine you have 2 fuel tanks, tank A and tank G. The pump that fills tank A only runs on fuel from tank G.
Okay, I like this analogy.
And the pump that fills tank G only runs on fuel from tank A.
That is a perfect analogy.
So if tank G starts to get full,
The engine roars to life and starts furiously pumping fuel into tank A to use up the surplus from G.
and vice versa.
Exactly. The surplus of one becomes the driving force for the production of the other. It is physically impossible to have a massive imbalance because the system is designed to be self-leveling.
It prevents a monopoly.
It prevents a monopoly. It's brilliant.
Okay, so we've built our A's and G's, but a factory that never stops running is going to, explode or at least fill the warehouse until the walls burst. We need to talk about Section 3, regulation, the brakes.
Feedback inhibition. It's the fundamental principle of metabolic control.
The product inhibits the process.
The more product you have, the slower the factory runs.
So if I have enough AMP and GMP lying around, they go back and tell the factory to, take a coffee break.
Precisely. And they do it at multiple checkpoints to ensure safety. Remember PRPP synthetase, the one that makes the scaffolds.
Step 1, the lumberyard.
That enzyme is inhibited by AMP, ADP, GMP, and GDP. Basically, if the cell is full of any kind of purine, it stops making the scaffold. It cuts off the supply of lumber.
Makes sense. Don't frame the house if you don't need a house.
Then remember the committed step? The big one.
The gatekeeper, PRPP glutamyl amidotransferase.
That's the one. It is strictly, stringently regulated. It is inhibited by both AMP and GMP. If the final products are present, the main gate to the factory closes. This prevents the cell from wasting glutamine and PRPP on a pathway that isn't needed.
So that's the main power switch. But what about that fork in the road? Does regulation happen there too?
It does. Excess AMP specifically inhibits the enzyme that converts IMP to more GMP.
Okay.
And excess GMP inhibits the enzyme that converts IMP to more GMP. This is a secondary layer of control. It prevents one branch from dominating the other, even if the main supply line is still trickling in.
So it's like a smart power grid. You can shut down the main power plant, or you can just shut down the substation that goes to a specific neighborhood.
Exactly. It allows for incredibly fine-tuned control over the whole process.
Now, we've mentioned how expensive this is. Energetically expensive, the text says over and over. So surely the body doesn't want to do this from scratch every single time.
No, it doesn't.
Which brings us to Section 4, the salvage pathway.
Yes. The green initiative of the cell.
Save the whales, salvage the purines.
In a manner of speaking, your cells are constantly turning over. DNA and RNA get broken down as part of normal maintenance. This releases free purine bases, adenine, guanine, and also hypoxanthine.
Hypoxanthine, that's the base form of IMP, the parent molecule, right?
Correct. It's the parent base, but without the sugar and phosphate attached. Now, the body has a choice. It can degrade these bases further and excrete them, which, as we'll see, is problematic, or it can grab them.
And recycle them.
It can grab them and reattach them to a fresh PRPP scaffold.
So it skips the whole 10-step, super expensive recipe.
It skips almost all of it. It's a simple one-step reaction. Purine plus PRPP yields a purine nucleotide plus pyrophosphate. Much, much simpler. And way more energy efficient.
It's the difference between building a car from raw iron ore versus just putting a new engine in an old chassis.
A great way to put it. And we have special recycling trucks enzymes to do this. There are two key ones to remember. First is APRT.
AP3.
Adenine phosphoryl transferase. It just salvages adenine back into AMP. Simple enough.
Okay.
And the second one is the big one, the really important one. It's called HGPRT. Hypoxanthine guanine phosphorybosal transferase.
HGPRT.
Got it. It's the workhorse. It salvages hypoxanthine back into IMP, and it salvages guanine back into GMP. This one enzyme is incredibly important for the body's purine economy.
And this is a bit of foreshadowing, right? Because you said earlier, if HGPRT is missing.
That's where we see Lesch-Nyhan syndrome. And we absolutely need to spend some real time on that when we get to the clinical section, because it reveals that this recycling isn't just about saving energy. For the brain, it seems to be about maintaining sanity.
Wow. Okay. So that's the purine story. Double ring, expensive to make, heavily regulated, and actively salvaged. Now let's flip the script. Section 5. Pyrimidine biosynthesis.
A completely different strategy.
How so?
With purines, remember, we built the ring on top of the ribose sugar.
Like building a house directly on its foundation.
Right. With pyrimidines, the cell builds the ring first, just floating freely in the cytoplasm, and then it attaches the finished ring to the ribose sugar later.
It's like a modular home. You build the structure in the factory, you truck it over, and then you crane it onto the foundation.
Precisely. And the chemistry reflects that. The whole pathway starts with a reaction catalyzed by an enzyme called carbamoyl phosphate synthetase II, or CPS2.
I noticed the 2. Is there a CPSY?
There is. CPSI is in the mitochondria, and it's part of the urea cycle. Its job is to clean up ammonia waste. CPS2 is in the cytosol, the main cell fluid, and its job is to make pyrimidine.
Different location, different job.
Different location, different function, and very different regulation.
So what does CPS2 use to start?
It uses CO2 and glutamine again. Very similar start. It forms a molecule called carbamoyl phosphate. Then, through a series of steps involving aspartate, we close the ring to form dihydroic acid.
And then we get to a molecule that sounds important, orotic acid.
Orotic acid is the key intermediate. It's the finished pyrimidine ring waiting for a home. It's your modular house sitting on the back of the truck.
And the home, the foundation, is PRPP again.
Correct. Orotic acid reacts with PRPP to form OMP, which is orodatine monophosphate. Then, in the final step, OMP is decarboxylated. It loses a CO2 molecule to become UMP.
UMP, uridine monophosphate. And that's the parent pyrimidine, right? Like IMP was for purines.
Exactly. From UMP, the cell can then make CGP for cytosine and eventually thymidine, the T in DNA.
And I notice the multifunctional protein theme comes back here too. We really seem to like bolting our machines together.
We do. The first 3 enzymes of this pathway, including CPS2, are all part of one giant polypeptide chain called CAD. It's a mega enzyme. And the last 2 steps, the ones that handle orotic acid and make UMP, are on another bifunctional polypeptide called UMT synthase.
So again, it's all about efficiency, no lost intermediates.
And the regulation parallels the purines too. CPS2 is the main brake for this pathway. It's inhibited by UTP, the final product.
So if you have enough UTP, you stop the production line.
Right. But interestingly, it is activated by PRPP.
Oh, that's cool. So if the substrate, the foundation, is piling up, it sends a signal to the factory saying, hey, we've got plenty of foundation over here, start building some houses.
Exactly right. It coordinates the supply of the sugar with the production of the base. It's a very slick system.
Okay, so we've made our purines, A, G, and our pyrimidines, C, U, T, but mostly we've been talking about the ribose forms, the RI and RNA. But the title of our deep dive mentions the blueprint DNA. So section 6 is about making the D in DNA. Deoxyribonucleotides.
This is a crucial distinction. The vast majority of nucleotide synthesis in the cell produces ribonucleotides. These are used for RNA, of course, but also for energy. ATP is a ribonucleotide.
Right. I always forget that. ATP isn't just some abstract energy battery. It's literally the letter A that goes into RNA.
Exactly. It's a dual-use technology.
Yeah?
Fuel and building block. But for DNA, we need the deoxy version. We have to remove an oxygen atom.
Deoxy, literally minus oxygen.
Yes, we have to reduce the 2' hydroxyl group on the ribose sugar. And we do this at the nucleotide level. We don't make a special deoxyribose sugar first and then build a base on it. We take the finished ribonucleotide diphosphate like ADP or GDP, and we strip the oxygen off.
And there's a special machine just for this one job.
A very special machine called ribonucleotide reductase. And this enzyme is only active when cells are preparing to synthesize DNA. That is, when they're about to divide.
That makes perfect sense. You don't need a stockpile of DNA building blocks if you aren't actually replicating your DNA.
Right. The mechanism is pretty complex. It requires a whole reduction system involving a protein cofactor called thioredoxin and an enzyme called thioredoxin reductase, which uses NADPH from the pentose phosphate pathway. It's basically a little electron transfer chain to perform this very difficult chemical reduction.
And the regulation here is described in the text as complex. Why so complicated?
Because the stakes are genetic mutation. To replicate your DNA accurately, you need a perfect balanced supply of DATP, DGTP, DCTP, and DTTP. If those ratios are off, if you have, say, 10 times more A than T, the polymerase enzyme that builds the new DNA strand will start making mistakes.
It'll insert the wrong letter.
It'll insert the wrong letter, and that leads to mutations. So this enzyme has a fascinating dual lock system. It has regulatory sites that control overall activity, whether it's on or off, and other sites that control specificity, which of the 4 letters it decides to make at any given moment.
So it's constantly adjusting its output.
Constantly. But the one big rule to remember, and this connects directly to the immune disease we'll discuss, is that DATP acts as a global inhibitor.
DATP turns the whole thing off.
If the level of DATP gets too high, it binds to that main activity site and acts as a kill switch. It shuts down the entire ribonucleotide reductase enzyme. It stops the production of all the DNA building blocks.
So if you have too much of just one letter, A, you stop making all the letters and cell division just stops cold.
That's exactly what happens.
Wow. Okay, listeners, keep that in mind. The DATP kill switch. Now let's move to section 7. We've built everything up. Now we have to tear it all down. Catabolism.
And this is where the story really diverges dramatically between purines and pyrimidines. One is, let's say, clean, and the other one is messy.
Very messy. Let's start with the problematic one, purine catabolism.
When you break down purines like AMP and GMP, you strip them down through a series of intermediates, inosine, hypoxanthine, and then xanthine. The final 2 steps are catalyzed by an enzyme called xanthine oxidase.
Okay.
And it converts xanthine into the final waste product, uric acid.
Uric acid, I definitely know that name. And it's usually not in a good context.
No. Uric acid is a problem for one main reason. Its solubility.
Oh.
Or rather, its lack of solubility. It is not very water soluble. And here's a weird evolutionary quirk. Most other mammals, your dog, your cat, they have an enzyme called uricase.
Uricase?
Yes. Uricase takes uric acid and converts it into a molecule called allantoin, which is highly water soluble. So dogs just pee out allantoin. No problem.
You don't have that.
Humans and the other great apes lost the gene for a functional uricase enzyme millions of years ago. We are, in this one specific regard, genetically defective. So we're stuck with uric acid.
So what happens to it?
Well, its pK is about 5.8. That means in acidic urine, it can easily form solid stones, kidney stones. And in the blood, which is at a pH of about 7.4, it exists as sodium urate. If the concentration gets too high, boom, it precipitates.
It turns into solid crystals?
Sharp, needle-like crystals. You can think of them as microscopic shards of glass floating in your tissues.
Ouch. And that is gout.
That is gout. Now,
Yeah.
compare that whole messy situation to pyrimidine catabolism.
The soluble one.
Exactly. When you break down uracil or thymine, you get CO2, ammonia, and 2 other molecules, beta-alanine and beta-imidazobutyrate.
And those are?
Highly water-soluble. They just float away harmlessly in urine. Or they're metabolized further into the citric acid cycle and used for energy. No fuss, no muss.
So there's no such thing as pyrimidine gout.
Exactly. You never, ever hear about people having uracil stones in their joints. Disorders of pyrimidine catabolism are very rare and don't cause obvious signs because the waste products just don't clog up the system.
The text did mention one interesting little blip about the pyrimidine waste, that beta-aminosobutyrate excretion increases during leukemia or after severe X-ray radiation. Why is that?
Because both of those conditions involve massive, massive destruction of DNA. Dead and dying cells release their DNA, the DNA gets broken down, the pyrimidines are catabolized, and you see a huge spike in this specific waste product in the urine.
So it's a grim signal. It's a chemical marker of large-scale cell death.
It is.
All right, we have laid the foundation. We know the factory, the breaks, the recycling plant, and the trash compactor. Now, let's go to the hospital. Section 8, clinical disorders and diseases. And I want to spend some real time here because this is where the biochemistry hits the real world. Let's start with gout.
Gout is the classic, the textbook disease of purine metabolism. The clinical term is hyperuricemia. Just means elevated uric acid in the blood.
And you said it's microscopic glass shards in the joints. Why the big toe? It's always the big toe in the cartoons.
That specific presentation is called podagra. And it often affects the big toe because it's a peripheral joint. It's far from the core of the body, so it's physically cooler than, say, your hip.
Ah.
And solubility decreases as temperature drops. So the sodium urate crystals precipitate out there first. It's just simple physics coming back to bite us.
So what causes it? Is it just eating too much rich food and wine, the disease of kings?
Diet certainly plays a role. Things like alcohol, red meat, shellfish are all high in purines and can trigger an attack. But biochemically, it's often an internal production problem.
Like what?
It can be overproduction of purines, maybe an overactive PRPP synthetase pushing the pathway too hard, or it can be underexcretion where the kidneys just aren't clearing the uric acid efficiently enough.
And the treatment logic for this is just brilliant. Allopurinol.
Allopurinol is a masterpiece of rational drug design. It's what we call a structural analog of hypoxanthine. It looks almost exactly like the real substrate for xanthine oxidase.
The enzyme that makes the uric acid.
Right. It's a decoy. The enzyme sees the allopurinol and grabs it instead of the real purine.
Yeah.
And here's the beautiful trick. Allopurinol is a suicide inhibitor. Once the enzyme grabs it, it gets stuck. It jams the gears of the enzyme permanently.
So you stop making uric acid. But wait, if you block the drain, don't you just get a backup of the stuff that comes before it, like xanthine and hypoxanthine?
You do. That's exactly what happens. But here's the magic. Xanthine and hypoxanthine are much more soluble in water than uric acid is. So by stopping the process one step early, you effectively convert a dangerous, insoluble piece of trash into a manageable, soluble piece of trash that you can just pee out without any problems.
That is so clever. You don't stop the trash, you just change the form of the trash.
Exactly.
Now let's move on to the condition that really stuck with me. Lesch-Nyhan syndrome. We mentioned the salvage enzyme, HGPRT.
Yes, this is a devastating disorder. And it's rare, but it teaches us something incredibly profound. It's an X-linked recessive defect, so it almost exclusively affects boys. And it's a complete or near-complete deficiency of the HGPRT enzyme.
So the recycling plant is closed for business.
Completely boarded up. And this has a terrible double whammy effect. First, you can't salvage hypoxanthine or guanine, so where do they go?
To the trash.
To the trash compactor. They all get degraded. This leads to massive uric acid production. These kids get severe gout and kidney stones incredibly early in life. Often as infants, you find these telltale orange uric acid crystals in their diapers.
That sounds painful enough, but you said that's not even the worst part.
No. The second consequence is metabolic. Because you aren't using a PRPP for the salvage pathway, the PRPP levels in the cell skyrocket.
Oh, and a high PRPP.
It's a powerful activator of the de novo factory. It pushes the whole synthesis pathway to run at full speed. So you're dumping more fuel on the fire, you have overproduction, and no recycling. It's a metabolic catastrophe.
And clinically, this leads to severe neurological issues.
Severe intellectual disability, spasticity, uncontrolled muscle movements. But the hallmark, the most characteristic and tragic symptom, is compulsive self-mutilation.
That's the part I find so baffling. We're talking about an enzyme that recycles a simple chemical base. How on earth does that translate to a behavioral compulsion where a child will uncontrollably bite their own lips and fingers, often to the point of severe injury?
That is the great unanswered mystery of this disease. These patients aren't doing it because they want to. They often scream in pain while they're doing it and will ask to be restrained. They are terrified of their own compulsion.
They're prisoners in their own bodies.
In a way, yes. It strongly suggests that purine salvage is absolutely critical for the developing brain, particularly for the basal ganglia and the dopamine pathways that control movement and impulse.
So a single missing enzyme in a recycling pathway can completely rewire the brain's behavioral control centers.
It appears so. We can treat the gout with allopurinol, but we still don't have a good way to treat the neurological tragedy of Lesch-Nyhan.
That is just heavy. Moving on. Von Gierke disease. I thought this was a glycogen storage disease, something to do with sugar.
It is. The primary defect is a deficiency in glucose 6-phosphatase. It's a liver problem. But remember, in metabolism, everything is connected.
Right.
If you can't release glucose from glucose 6-phosphate, that molecule gets trapped and shunted into other pathways. One of those is the pentose phosphate pathway. And the major product of that pathway is ribose 5-phosphate.
Which is the starter molecule for purine synthesis.
Exactly. More starter leads to more PRPP, which leads to more purines, which leads to more uric acid, which leads to gout. So you get gout as a secondary complication of a primary problem with sugar metabolism.
Everything's connected. Next up, adenosine deaminase, or ADA deficiency. The so-called bubble boy disease.
This is a cause of severe combined immunodeficiency, or SCID. The defect is in the enzyme adenosine deaminase, which, as the name suggests, handles the breakdown of adenosine.
So if you can't break it down, it accumulates.
Adenosine accumulates, and it gets phosphorylated by other enzymes into deoxy-ATP.
And wait for it, the kill switch?
You remembered. High toxic levels of DATP build up in the cell, and they bind to ribonucleotide reductase and shut it down.
So the cells can't make any of the other DNA building blocks.
Correct. And which cells in your body need to divide rapidly and constantly to do their job?
Your immune cells, T cells and B cells.
Exactly. Without the DNA precursors, they need to proliferate and mount an attack. They just can't. The immune system effectively vanishes.
That is a brutally clear chain of events. One missing enzyme leads to high DATP, which blocks the DNA factory, which means no immune cells.
It's a classic example of metabolic toxicity. The buildup of one molecule poisons another, essential process.
Lastly, in the clinic, orotic aciduria.
This one is a great illustration of the pyrimidine pathway. It's a deficiency in that bifunctional enzyme at the end of the line, UMP synthase.
So you can make orotic acid the modular house, but you can't attach it to the foundation. You can't make UMP.
Exactly. You can't convert orotate into UMP. So orotic acid, the precursor, builds up and up and up. You see these characteristic orotic acid crystals in the urine.
And what's the clinical effect?
Well, if you can't make UMP, you can't make CTP or TTP. You can't make DNA. This leads to a failure to thrive and megaloblastic anemia. The bone marrow can't make red blood cells properly because the cells can't divide.
And the treatment for this?
It's surprisingly simple and elegant. You feed the patient uridine.
You just give them the product they're missing.
You give them uridine. It can be salvaged and phosphorylated to UMP by other enzymes, completely bypassing the block. It solves the deficiency, and what's more, the newly made UMP and UTP then provide feedback inhibition to shut down CPS2, which stops the overproduction of orotic acid.
Wow. Amazing. Sometimes the solution really is just give them what they're missing.
It works beautifully in this particular case.
Okay, finally, let's wrap up with Section 9, pharmacology. We can use our intimate knowledge of these pathways to fight the ultimate disease of cell division, cancer.
Cancer cells are, by definition, addicted to nucleotides. They are voracious consumers. They need to replicate their DNA constantly to fuel their growth, so we starve them.
Let's talk about methotrexate. That's a very famous chemotherapy drug.
Methotrexate is a structural analog of folic acid. It competitively inhibits the enzyme dihydrofolate reductase.
So it stops the regeneration of the folate donor, the one carbon delivery truck.
Correct. Without the active tetrahydrofolate, you can't donate those two crucial carbons to the purine ring. And this is key. You also can't make thymidylate, the T for DNA. The factory runs out of essential parts. The cancer cell tries to divide but fails.
And what about 5-fluorouracil, 5-FU?
That's a pyrimidine analog. It looks just like uracil or thymine. It specifically and powerfully inhibits the enzyme thymidylate syntase.
The one that makes the T.
It stops the conversion of DOMP to DTMP.
So it specifically blocks the T in DNA.
Exactly. It's a phenomenon called thymineless death. The cell tries to replicate its DNA, it realizes it has no Ts, and it triggers apoptosis. It dies.
For brutal. And 6-mercaptopurine.
That one's a purine analog. It gets
Yeah.
converted in the body to a nucleotide that inhibits the conversion of IMP to AMP and GMP. It locks that fork in the road we talked about earlier.
So all of these drugs are essentially just throwing sand in the gears of the specific enzymes we just toured.
Yes, they are rational attacks. They target the bottlenecks, the folate supply, the thymine conversion, the IMP branch point. They exploit the cancer cell's desperate need for speed and its reliance on these pathways.
Well, we have covered a tremendous amount of ground today, from the liver's assembly line to the tragedy of Lesch-Nyhan syndrome to the life-saving mechanics of chemotherapy.
It is a very dense landscape. but it's the landscape of life.
So let's just try to summarize the journey. We start with simple atoms glycine, CO2 from the air we breathe out. We invest a ton of ATP to build these complex double and single rings.
A huge investment.
We have to balance them carefully, A versus G, purine versus pyrimidine, using these elegant cross-regulation systems.
Right.
We have to recycle them with salvage pathways because they're just too expensive to make from scratch every time. And when we're finally done with them, we have to dispose of the waste carefully, or else we get microscopic shards of glass in our joints.
That is the life cycle in a nutshell, the blueprint and the waste.
I want to leave our listeners with a final thought to chew on. We touched on it with Leishnahan. We usually think of metabolism as plumbing, pipes, flows, blockages. But here we have a broken pipe, a single missing salvage enzyme that leads directly to a child hurting themselves against their will.
It's a profound connection.
It suggests that these molecules, these purines, are doing something in the brain that is so fundamental to our behavior, to our sense of self-preservation, that when their levels are off balance, our most basic instincts can get horrifyingly cross-wired.
It really raises the question, how much of us, our personality, our choices, our behavior, is dictated by the rate of flux through a metabolic pathway? At the end of the day, we are chemical beings.
A sobering and absolutely fascinating thought. Thank you for taking us through the intricate chemistry today.
My pleasure. It was fun.
And thank you to our listener for diving deep with us. We hope you'll never look at a piece of steak or your own big toe in quite the same way again. This has been The Deep Dive. We'll see you next time.